Good morning, everybody. I think we will start. It's 3 minutes past 10, and I think there are so many webinars today that we cannot lose the time or have such a delay in with our webinar. My name is Stephanie Siebert. I'm the Executive Director of ECN, and I'm happy to celebrate with you the World Soil Day 2023. First, some general rules. Please keep your microphones muted. We will have 2 presentations today, and after each presentation, you have also the possibility to pose questions. Further, I would like to let you know that we will record the meeting so that we have also the possibility to place the webinar afterwards on our YouTube channel. Yeah, that's for the time being. I'm now going to chair our presentation. Okay. Our webinar today is about Europe's new soil monitoring law and compost effect on soil biodiversity. We are very proud and thankful that the University of Padua has given us its patronage and that the Department of Biology supports our webinar by presenting its latest research results on the effect of compost on the soil biota. The World Soil Day 2023 is titled Soil and Water, a Source of Life. Just by listening to these words, it is clear that soil and water are, besides air, the most valuable resources for life on Earth. Soils deliver functions which are fundamental to soil fertility, plant nutrition, and water filtration. Water is essential for all living organisms, humans, animals, plants, and microorganisms. Both resources are under threat. 60 to 70% of European soils are unhealthy, and access to high-quality drinking water is still limited and will become more and more challenging. The European Commission has tackled these soil threats as listed here in the Soil Strategy for 2030. With the publication of the proposal on a soil monitoring law, published this summer on 5th July, we welcome that the Commission has tabled back soil as an essential source on the political agenda. Today, with the webinar on the World Soil Day, we would like to take the chance to introduce to you the new legislative approaches on soil in the European Union and stress the importance of healthy soils. Soil organic matter and sustainable soil management practices play a key role to keep our soil healthy. Applying compost and digestate as organic soil improvers and fertilizers can contribute to this. But I do not want to spend too much time, and I want to introduce the first speaker of today. We will start with a presentation by my colleague, our ECN policy officer, Riccardo Cambini, who is responsible for environment and agriculture in ECN, and he is actively following the legislative developments at European level. Riccardo, the floor is yours. I will stop sharing and you can start to share your presentation.
Thank you, Stephanie. Good morning, everybody, and thank you for being with us today. So let's start sharing my presentation. I hope everybody can see it. I will skip the introduction since Stephanie already did this. I just want to quickly introduce who is ECN, but I will not spend too much time. So the European Compost Network is an organization who has members all over Europe, actually 67 members. And as you can see, our mission is advocating for the separate collection of organic waste and turning into high quality compost and digested for soil health. So we are very committed for having healthy soils in Europe and more in general all over the world. At the core of what we do, circularity and sustainability are the principles that lead our actions. I just wanted to, before delving into the new proposed soil law, to give a little bit of context because it's important to understand better where we are now. The European Commission back in 2019 has published its strategy for the next 5 years. We are towards the end of the 4th year, and the strategy for the green transition is the European Green Deal. And within the European Green Deal, there are included a lot of other strategies where bio-waste plays a key role, as like the bioeconomy strategy, the biodiversity strategy, but also other action plans for circular economy and zero pollution. So it's important to know that bioeconomy is very present in the plans of the European Union to achieve climate neutrality in 2050, which is the end goal of this European Green Deal. If we zoom in, we have the biodiversity strategy, and a new vision for soil is anchored in this biodiversity strategy for 2030. And together with it, in 2021, the European Commission has published the soil strategy. And the soil strategy foresees both medium-term objectives and for 2030 and long-term ones. And the main vision for this soil strategy is to have all soil ecosystem in healthy conditions by 2050. And these medium targets also address different challenges and issues linked to soil, such as pesticide reduction, nutrient loss reduction, desertification, and also water quality and chemical status. So this is all intertwined with soil. And if we zoom in a little bit more, what does the soil strategy say? So the soil strategy sets key main challenges and how to address them. And we have of course, climate change, soil biodiversity, water resources, but we also have soil and the circular economy, which is super important. And in this connection between soil and circular economy, we see that there is the need to close the nutrient and carbon cycles through the recycling of organic matter with compost and digested application, for example. So this is also one of the things where the European Commission has committed itself to strengthen its actions. We as ECN have published our feedback to this soil strategy, and so we have contributed with our inputs, and we supported our stance with some results from long-term field trials. Together with the Italian Composting Association, so that also the Commission recognized the importance of the application of high-quality compost and digested in soil for soil fertility and health. To go more into the details, before I think there's somebody has his mic open. Please, if you can mute yourself. So if you want to go a little bit more into the detail before, this is the path towards the adoption of the proposal for a soil monitoring law. It was a bit long, as you can see, more than 1.5 years after the publication of the soil strategy. Yeah, there were some challenges in the middle of the process, but finally, and with some delay, finally the Commission has published its proposal within the package of the sustainable use of natural resources that included other different proposals. So to go into the content of the soil monitoring law, we have 6 main different blocks, let's say. Of course, the subject matter, which the directive applies to all European soils, and the scope is to set common monitoring and assessment measures, and also to tackle sustainable soil management and contaminated sites. The Commission, although the vision for soil included in the soil strategy did foresee mandatory targets for 2050, in the end, the Commission decided not to include mandatory targets for 2050 to reach all soils in healthy conditions. And then we have, of course, a definition as usual, and the establishment of soil districts in the European Union, and the designation of competent authorities that would deal with these soil districts. And then the most important parts are of the monitoring and assessment chapter, that I will go more into the details in a bit. But to give you a preview, this part sets soil indicators and criteria, so thresholds or limit values to the different type of soil degradations, and also sets rules for sampling methodologies, and importantly, it includes this one out, all out principle, so that if one of these soil criterion is not met, then the soil will be labeled as not healthy. So remediation practices should be included. And then we have sustainable soil management, which is very important. There is the definition of sustainable soil management practices and the land take mitigation principles. And here we will also see it in a bit. There is the obligation for member states to set up practices respecting principles that have been laid down in this directive. After we have a whole chapter on contaminated sites, which is a little bit more of a lex specialis, let's say, because it tackles a specific issue for soils, which is soil pollution, and it sets a risk-based approach to identify, investigate, and manage contaminated sites. Also, envisioning a national public register for each member state. And in the end, we have a part on financing and access to justice, which is very important, that the participation for citizens and public is included in case of failures of implementation of this directive. So yeah, on sustainable soil management, the member states are obliged to define sustainable soil management practices respecting sustainable soil management principle that are listed in the Annex 3 that goes with this proposal. And if we look more thoroughly at these principles, we can see that one of them is focusing on the prioritization of circular solutions for when fertilization is applied, so to increase the organic content in the soil. And this is, of course, particularly important for the application of compost and digest. That this was really made for these secondary raw materials to incentivize their use. And if we keep on looking more in detail, there is this part on soil descriptors that has been defined by the European Commission in 4 different parts. The first part includes soil descriptors or indicators for which criteria have been established at common European level. So all the member states have to abide to these descriptors and criteria. And then the second part, where the soil descriptors are established at national level, with of course some indications from the Commission on how to set this. And then a part with soil descriptors that do not have criteria and are where their monitoring is optional. So it's not mandatory. And a last part on the land take and soil indicators specifically. And I just want to give you a little snapshot of the different aspects of soil degradation that are included. Maybe for you to see if they're both exhaustive or not. But here we have, for example, the indicators that are established at union level. We have salinization, soil erosion, loss of soil organic carbon, and some soil compaction. And yeah, on the right column you can see the descriptors, and then you have the criteria and also some exceptions to the monitoring and the assessment of these aspects. For example, if the soil is already saline for natural conditions and reason, then the soil would be exempted from the monitoring of salinization, just to give you an example. And then we have other indicators set at member state level. So for this, the member states will have to find a good criterion or limit value or threshold. We have phosphorus content, soil contamination, and water holding capacity. And for the optional monitoring, we have these 4 indicators: nitrogen. Among them there is nitrogen and also soil biodiversity, for which they have included different optional descriptors. And so this is a little bit the timeline for the soil monitoring law. This is still a proposal, and I will tell you a bit more on the legislative process. But yeah, the first assessment of soil health will be in 5 years from the entry into force of the directive, and then a first evaluation of the directive will come after 6 years from the entry into force. ICN has published its position after the adoption of this proposal, because the Commission always opens a feedback period on its initiatives, and our main focus was on this particular issue. So we asked for the inclusion of binding targets for 2050 to track the progress of member states. And we advised the institutions to set soil descriptors that would work synergistically together, and prioritize the sustainable soil management practices and make their implementation more swift from member states, and then support soil health certificates, which are included in some of the provisions, but with social safeguards in place for land managers. So this was very important. So at this time we are at the heart of the co-decision process. So the European Parliament and the Council are working on the file to propose their amendments and then to vote them. I wanted to flag just some of the peculiarities of the European Parliament proposal and some of its amendments, which still have to be voted. So instead of a one out all out approach, which I mentioned before, the European Parliament is proposing a more nuanced approach to assess soil health, which is called the soil ecological status approach, by setting a classification of soils. And if the classification, or if the soil classifies among the two best positions, then it's considered healthy. And if not, the member states have to take action to remediate and improve the classification of the soil. And you can see if for critically degraded soils, the timeline is for 10 years, and for degraded and moderate ecological status of soil is 6 years. It is not said whether they have to reach healthy status or they have to move up in the ranking of one level, for example. This is done on purpose to leave the flexibility to member states, but it's a little bit unclear also. So, and then we have other amendments from the European Parliament that has proposed, of course, binding targets, as also ICN has asked for, and many other organizations. And then it is encouraged to establish soil district plans to set vision and measures applicable to each territory and achieve the 2050 target. And then on sustainable soil management, there is this innovative proposal from the European Parliament with the introduction of a digital platform which is public and where all the stakeholders can and the land managers can share their best practices. I come to the conclusion. So this is a little bit what to expect for the next few months. The law is still undergoing revision process, let's say, and it will probably be agreed in the next term, but it will be important to have a position from the European Parliament and the Council of the European Union, which are foreseen for April and May next year, and then the final negotiations will start to adopt the final text. Thanks a lot. Here there are some websites, and if you want more information, you can always write. Thanks a lot.
Thank you very much, Ricardo, for giving us a comprehensive overview about the new proposal of the Commission and about the political discussion. We have some minutes over for some questions. Are there any questions from the participants? Then please raise your hand. Okay, I do not see any hand, but probably if you have any question later on, you can also send us an email.
Think
we can.
Jane has a hand.
Oh, okay, I haven't seen. Okay, Jane, then go ahead.
Good morning. I might have missed this, but are there any proposals for monitoring plastic contamination, and in particular microplastics in soils?
So this is left to member states, according to the Commission proposals. This is left to member states if to include other contaminants, because they have set a list of heavy metals to be monitored, but they are leaving this flexibility to member states if to include other pollutants and contaminants in their monitoring. But the European Parliament, in the European Parliament draft amendments, there is the let's say that this has been strengthened. So they want to come up with a watch list also to monitor emergent pollutants and contaminants of greatest concern, and microplastics is one of them. So we still have to see in the final negotiations what will be agreed. But the European Parliament has been a little bit more ambitious in this regard, at least on the monitoring side, because and then for the assessment and the eventual development of thresholds or limits values, we will have to see in few years after the evaluation of the directive.
Okay, thanks.
Okay, I don't see any further hand. So we really will follow the discussion in the Parliament and as well in the Council, and we hope as well that they come to a compromise at the final end, so that the negotiation will start next summer after the new election of the European Commission, and that this proposal by the Commission will not be blocked by any party in the European Parliament as it has happened in the past. Okay, now we are going to the second presentation, to our second speaker. I'm very happy to welcome Sebastian Nigris. He is a researcher in the Department of Biology of the University of Padua, and he's working in the research lab of the Botanical Garden of Padua. He has obtained a PhD in 2015, and since then he's working on soil microbiota and on the interactions between plants and soil bacteria. During his career, he collaborated with the Wageningen University and Research in the Netherlands and with the New York Botanical Garden. Recently, and he will present today us his results, he has started a scientific collaboration with CESA. This is an Italian company producing compost to investigate the effect of compost on the soil and plant-associated microbiological communities. Sebastian, it's now your turn to present your results.
Okay, thank you very much, Stefan, for the nice introduction. Shall I share the... Okay, I think it's this one. Yep. Okay. Can you see my presentation?
Yes, we can. It's perfect.
Okay, so good morning to everybody. It's really a pleasure for me to be here virtually to talk about my a part of my research about the effect of on compost on soil and plant microbial biodiversity. So firstly, I wanna spend a couple of words about my research group that is actually headed by Professor Barbara Badan, that she's connected in to this presentation. And we are actually botanists, so it's a research group that has a long experience on the study of interaction between microbes and especially to the molecular mechanism underlying these plant microbe interactions. And so as botanists, we are really interested to understand how plants can interact in soil with microorganisms, because actually a great part of the plant biomass is actually below the soil, is below the ground in the soil, and plant roots have to interact not only with the physical component of the soil that is super important, like nutrients, water, mineral nutrients, and organic matter, but in the soil, the plant have to interact with a huge amount of microorganisms, especially fungi and bacteria. And some of these interactions are actually pretty popular and well known, like in the case of the arbuscular mycorrhiza interaction between fungi, arbuscular mycorrhiza, fungi, and plants, and the interaction between rhizobia and legumes to form the nodules for the nitrogen fixation. And all these interactions are well studied, all the molecular mechanisms are known, but are limited to a restricted number of species, both for the microorganisms and for the plants. But there are many interactions in the soil between the roots of all the plants living in the soil and a large plethora of bacteria. And these interactions are less studied, and we actually don't know all the mechanisms that are subtending these kind of interactions. So around the root, there is a small portion of soil directly in contact with the roots that we call rhizosphere, and all these bacteria that can live there, that actually colonize this rhizosphere and the root surface, we can call them rhizobacteria. So it's a specific group of bacteria. And there is a decreased number between the soil bacteria, that is the main source of bacterial group that can interact with plants. The number of bacteria, these are some rough estimation of the number of bacteria that we can find in the rhizosphere, and then these bacteria can also enter in the tissues of the roots, colonize really the inner tissues of the roots, and there is a decrease in number of these species and the number really of bacteria that can interact with roots, because there is an increase in control by the plants to select some particular species that are useful for the life of the plants. So which are the effects of this interaction? We can recognize some useful effects of these rhizobacteria for the plants, because they can improve the nutrient uptake by the production of molecules that allow, for example, the binding of iron ions and the uptake by the plant of iron. They can solubilize phosphate. They can convert right there, so super close to the root. They can convert nitrogen from different chemical species to other that are more available for the plant. And so with the global effect to improve the plant biomass production, that is also improved by these bacteria because they can produce molecules that are similar to plant phytohormones. So they directly stimulate the root proliferation and the production of biomass. So they also have a big impact on plant health, directly controlling the pathogen presence by biocontrol activity that is performed by producing antibiotics, and these let's say beneficial bacteria can compete for space and nutrients with other pathogens, with soil pathogens, and they also can induce a systemic response of the plant that actually prime the plants to face pathogenic attack. And so they can really respond in a better way to pathogens. So this global effect of an improved nutrition, a better production of biomass, and a better health for the plant, it's actually a way to control also stress for plants. So they give a kind of protection from biotic and abiotic stresses to the plant. And we all know that plants are facing to big changing of the environment. So it's a way to improve the resilience of plants in a changing environment. So many factors actually influence the plant microbe interaction. It's not a random process. So the main driving factor is the genotype of the plant. So every plant species can select a group of species, of bacteria species that are allowed to interact with the roots, and also the developmental stage and the stress level of the plant. So stressed plants can recruit different bacterial groups to cope this particular stress. And of course, the soil characteristics and the soil microbial community, that is the main source of bacteria that can interact with plant. It's important to shape the community that is able to interact with plant. So it's easy to understand that if there is a high microbial biodiversity in soil, the plant has more chance to find in the soil the beneficial taxa of bacteria to interact with. But actually, we are facing a big problem that we already talked about today. That is that the soil biodiversity is actually declining. So a lower level of soil biodiversity is also a problem from plant health and productivity. So there are different causes of this loss of biodiversity in soil that are the use of chemical fertilizers and pesticides in agriculture, heavy tillage of the soil, so a deep laboration of the soil, and the focus on monocultures. And fortunately, we have a bunch of possible solutions that we can apply to sustain, to protect the soil biodiversity. That is crop rotation, so different species during the time can improve the soil biodiversity. A shallow tillage of the soil, and the use of organic and biofertilization. So you probably know better than me what compost is. It's and it's rich of organic matter, and it has been used since a lot of time to improve the structure of the soil, to give nutrients, and to improve the structure of the soil with a lot of organic matter. But we also considered the compost as a source of microorganisms because it's a complex matrix that has a complex microbiota, a unique microbiota. So our research question was if the compost can really enrich the soil bacterial community, and moreover the rhizosphere bacterial community. So to answer this question, we built a network between the Department of Biology and the SESA company, that is a compost producing company in Italy, and with the support of the New York Botanical Garden, when I spent some months to work there on this, also on this project. And to answer to our question, we set up quite large experiment, use tomato plants as model plant, as model species. We set up many replicates to have a good statistic strength, and we use a set of plants fertilized with compost and a set of plants fertilized with sterile compost to kill all the microbiological component of the compost as a control. And another control, we fertilize plants with a chemical fertilizer to mimic the traditional agricultural approaches. And we sample from these plants, we sample the bulk soil, that is the soil far from the roots. We sample the rhizosphere soil, so the soil that was intimately binded to the roots themselves, and the soil and the compost as a reference, as a control. So from these samples, we extracted the DNA because we use a DNA-based approach to describe the communities of bacteria related to these samples. So first we extracted the total DNA for each sample, and then we isolate bacterial DNA, and then we sequence it. And the sequences have been analyzed by statistical software to describe the community of every sample that we considered, and we wanted to compare. So the first analysis that we have done is a beta diversity analysis, and this analysis allowed us to see if the bacterial community of different samples could be actually different among them. So what we have seen is that the community of the rhizosphere of the plants that have been treated with compost is actually different from the rhizosphere community of plants that have been fertilized with a chemical fertilizer. So this result actually it can be intuitive. So if I put a complex matrix in the soil, I can modify the biological structure or the biological components of the soil. But with our data, we gave a robust statistical significance to this result. So we can claim that the use of compost in our experimental setup led to a different bacterial community for the rhizosphere compared to plants that have been fertilized with a chemical fertilizer. So this result says that the two communities are different. But how these communities are different? Did we improve the community using the compost? And to answer these questions, we perform an alpha diversity analysis that is a tool to investigate the biodiversity and the richness of species within a sample. So how many species there are in one sample compared to the others. And what we have seen analyzing different ecological indexes is that the rhizosphere of the plants treated with compost has more species than the rhizosphere of the plants treated with a chemical fertilizer. So if we use compost, we actually improve the rhizosphere biodiversity of the plants treated with compost. So this is a big result because, as I said at the beginning, a higher biodiversity means a bigger chance for the plant to find beneficial bacteria. And to understand which are the bacterial groups that are involved in this enrichment, we perform also qualitative community analysis. studying the relative abundance of the bacterial groups that we found in the different communities. And this is just an overview of all the samples that we analyzed. And I wanted to focus on the rhizosphere again, because it's the most crucial environment, let's say, for the plant health. And what we have seen is, for example, that in the rhizosphere of the plants treated with compost, we have an enrichment of the Bacillus species belonging to the genus Bacillus. And you can see here, let me show on the screen with the LAS. Okay, you can see here, this is the description, let's say, of the community that we found in the compost. And you see this blue section. This group of bacteria is assigned to the Firmicutes, so is the phylum in which belongs the Bacillus species. And you see that in the compost is super enriched. And you see that also in the rhizosphere of the plants that have been treated with compost is, let's say, larger than the blue section of the community, in the community of plants treated with a chemical fertilizer. So we hypothesize that there is a direct transfer from compost to the rhizosphere. And this group is particularly interesting and useful for plants, because among the Bacillus species, there are a lot of species that can positively interact with the root, and these interactions have beneficial effects for the plant. So they can help the plant to cope against drought stress, salt stress, cold stress, but also, and most importantly, heat stress. And we know that our agricultural systems are experiencing heat waves even more frequently along the years. And these species, so species belonging to Bacillus, can really interact. We know that they can also enter in the root tissues in a beneficial way, and they can protect the plant also from pathogens. So it's a way to make the plants more resistant to new arising pathogens. So with our data, we have seen that actually the rhizosphere of plants that we treated with compost actually hosts more species. And this is said by the alpha diversity analysis. And we know that with a statistical, we can be confident, because the statistic says that the bacterial community is also significantly diverse. And we saw it also in qualitative terms, so because there are some specific taxa that are enriched in the rhizosphere of plants treated with compost. So we can conclude that compost can really influence soil, because from soil these taxa can interact with the plant and finally enrich the rhizosphere microbiota. And as I just said, the Bacillus species play really a critical role in plant protection, and we demonstrate that compost is not only enriched in Bacillus species, but these species can be transmitted to the plant rhizosphere. So, of course, this is just the first part of the research. It's just one aspect, because we are doing much more, because we are using also an isolation approach to get the bacterial species, the culturable bacterial species that are present in the compost, to characterize them and to test which beneficial properties they could have to help the plants in a single way, and to perform synthetic communities to add to the compost to create, like, how can I say, improve biofertilizer. And beside this, we are also investigating the molecular mechanism that underlie this interaction and underlie the effects on the use of compost on plants. So we are performing in vitro and in vivo growth tests using tomato as model plant for the horticultural species, and we are trying to assess also the morphological effect of the use of the compost, of the use of these isolated strains and the compost and bacteria together to identify the mechanism that can improve the root biomass, the root nutrition, also by analyzing root morphology and by the analysis of gene expression when the plants are fertilized with compost, when the plants are matched with these beneficial bacteria. And we have 2 PhD students working on it. And maybe the next time we will present more, also some molecular data that I think are important to describe this mechanism of the beneficial effects of the compost on the plant cultivation. So we can discuss some general conclusions about the use of the compost in agriculture. So compost is known to be a source, right, of organic matter it's useful to improve the soil structure, but also we demonstrated that the compost can also enrich the soil biodiversity, and as a consequence, the rhizosphere microbiota that is super important for plant health, for all the aspects that I presented you. So we can conclude that the use of compost is actually a valuable and sustainable strategy because it's a product of circular economy, right? To reduce the impact of agriculture on soil biodiversity. So the improving of soil biodiversity is a smart way and sustainable way to improve plant health and to sustain plant production in a changing environment. So there are many people working on this project and would like to present them. So we have a couple of PhD students, Rosalba Cipriani and Giulia Ghirardello, a master student that has just started to work in this project, Leonardo, Professor Barbara Baldan that is heading the group. We have a couple of former students, brilliant former students that actually perform all the research. And we also are collaborating with Professor Nicoletto from the Agricultural Department of our university. And I want to really thank the people from the SESA company, Dr. Verne Zanardi and Dr. Tiziano Bonato, that are that we are collaborating in a super productive way to discover these new aspects of the use of the compost in agriculture. And of course, I thank you for the attention, and I'm happy to answer to all the questions you can have.
Thank you very much, Sebastiano, for your great presentation, for presenting these valuable results for our sector. And it was also for me very interesting to hear about this, that you have found out that the soil biodiversity is increasing when applying compost. When I made my PhD study, we only looked into microbiological activity and enzyme activities at this time at the beginning of the 19th. It was not usual to do the DNA analysis and to find out the specific species. So these are really good results, and I hope that we also will get further information from your research in future. And thank you as well to your team. This is very promising. But now I would like to give the floor to the participants if there are any questions to your presentation. Then please raise your hand.
Yeah, I saw a couple of raised hands.
I do not see any.
I raise my hand. If I can take the floor.
Yes, yeah, for sure.
Yeah,
Ben.
hi, good morning. This is Agnese Boccalon from ACR Plus. So thanks, Sebastiano, for the presentation. I have two questions. One, if you did the testing on different soil types, or if you applied the testing only on a specific soil type, and if you did it on different ones, if you found some variation in the amount of bacteria present in the rhizosphere. And the second question actually relates to a slide where you were showing the different composition of the bacteria present. So I don't know if you can get back to that.
Yeah, sure.
Uh,
Yeah, that one with all the samples, or just this
yeah,
one with the
this one. Yeah,
rhizosphere?
this one.
Okay.
Yeah. So I was wondering, in the second last column, that is the application of the chemical fertilizer, right?
Yes.
So what is the red part?
Proteobacteria.
And because I don't know the function of the protobacteria on soils, but that, let's say, is higher than the compost one. So I don't know if you can explain a bit what's the impact. Thanks.
Okay, related to the first question, we just tried we prepare like a mock soil that is close to agricultural soils, making a mix of different components, and we chemically characterize the soil, and the physical characteristics as well. What we want to do now is to transfer this type of analysis in a real agricultural system. And that's why we are collaborating with the Department of Agronomy, because they have experimental fields in which compost has been used since years. So we are thinking to perform a long-term analysis to see the long-term effects on the compost use on the rhizosphere. So for the moment, this is like a controlled situation and to test the effect in a small scale, but we are thinking to enlarge the scale using a real agricultural system. So that's the first question. The second question, that's a good question actually, because I highlight a result in which the compost actually enriched a group of bacteria. But of course, there are other differences. Actually, Proteobacteria is a diverse group of species, so there are many species, and still some of them have beneficial effects on the plant, and others are just soil bacteria. So it's difficult to tell if they have. It's not specific like the Firmicutes that is mainly composed by Bacillus, and we know a lot about Bacillus. About Proteobacteria is less. This phylum of bacteria is less characterized, let's say, and they have the components, the species belonging to this phylum are less related to beneficial effect on plants, let's say. Okay.
Thanks.
Okay, I saw a second hand from Penelope.
First of all, congratulations for your research. And I would like to know which kind of compost did you use? What it was made of?
Okay, is deriving from the food scratches collected from the municipality of Padova, and with a portion of plant material. So I don't know the real composition. I mean, I know their composition, but I don't remember the percentage of waste from food, from urban environment, and from green material. But is produced firstly by anaerobic fermentation to get biogas, and then with anaerobic fermentation to produce the final compost, let's say.
But it could be something that influence in the results of the microbiota of the soil.
Well, it could be interesting to compare different composts produced by different companies with different producing systems. I'm not super expert about the production system and the let's say, industrial characteristic of every kind of compost. But of course, if you change the composition of the initial of the starting community that you apply to the soil, of course, that can be a change in the final result. But I think that I think the production is quite standardized, so I'm pretty sure that every kind of compost has a own microbiota that can interact with the microbiota of the soil.
Okay, thank you.
Yeah, perhaps I can complement to this. Normally, the system in Italy is door-to-door collection of food waste from households, so food residues from the kitchen cooking. Then it goes into anaerobic digestion plant for producing biogas, and afterwards it is composted by adding green waste structured material. And normally you need as a minimum 20% of structured material to be added for a composting process to get air into this. So it's separately collected bio-waste from households mainly. I see as well another question in the chat from Christoph Garas from Finland. He's asking if we can expect the same effects also from digestate. So if we only have an anaerobic process, not having a composting process. Sebastian, do you have any experience on this or on the communities? The microbiological communities differ probably under anaerobic conditions and under aerobic conditions.
Yeah, no, actually, I don't have any experience about the use of the direct use of digestate. I know that there is a big debate in Europe around the use of this material directly as a fertilizer, and of course, every material in which there was a microbial-driven transformation as a microbial community. So probably, as you said, the microbial community of the digestate is more enriched of anaerobic strains rather than the compost that has been treated in an aerobic way. So for plants, probably is more useful the use of material that had an aerobic phase during its production to enrich the material in species that can use oxygen, because soil is actually an environment in which oxygen is present.
Okay.
But yeah, it's something that can be studied. I mean,
yeah. But thank you very much. I don't see any further hand raised, and I think we have discussed the topic which compost was used. These were also 2 additional questions in the chat. So thank you again for giving your presentation, to presenting your first results, and I really am looking forward for the final results when they are published, and also your further research. This is very, very good. We know that
maybe we can share. Yeah, we can share the publication and when it will
that would be
out.
great.
Yeah.
So thank you very much for this, because we know that the soil biodiversity was always a little bit overlooked for a long time, but we hope that with this webinar, with this presentation of these results, but also with other presentations which were given also by the Global Soil Partnership under the FAO, and also the work of the Coalition Action for Soil Health. They are also bringing forward soil biodiversity into the awareness of for us. I will share my presentation again, because we are coming to the end of our webinar today. I hope you can see it. I think so. Okay. We all know that to keep our soils healthy is of utmost importance, and we are really happy that the Commission has taken a first step to take awareness or raise awareness on soil health with the new proposal. And it's now up to the political decision maker to bring forward a robust soil monitoring law with the goal to reach the overall sustainable goals that our soil ecosystems should be under healthy conditions by 2050. But we, as human beings, we can and have to contribute to this as well by supporting the protection of soils with sustainable soil management, but also with our nutrition and also to change our lifestyles. I think there's time that we have to consider this. As we heard today, organic matter plays a fundamental role for soil well-being and consequently for us humans. Still in 2019, we have, together with our Italian organization SCIC, we have introduced this initiative Safe Organics in Soils. And with this initiative, we want to promote, or we make awareness for healthy soils and as well for increasing soil organic matter in our soils. We had heard at the beginning that our soils are lacking on organic matter, and we see that with our products of compost and as well of digestate, we can increase and maintain the organic matter content in soils. So if you want to support our initiative, please go on our website Safe Organics in Soils and sign our manifesto. Finally, I would like also thank again the University of Padua and the Department of Biology for the contribution today, and as well I would like to thank as well my colleague Riccardo Cambini for presenting the new soil law. But the day is not finalized, and you can also have a look on our website for further information, as well on our position paper on the soil monitoring law, but also on the soil strategy. And we have as well a regular newsletter where you can also subscribe in. So visit our website on www.compost-digestate.eu. But today is the World Soil Day, and I would like also to invite you to celebrate the whole day with webinars, because we are interacting with the International Compost Alliance. The International Compost Alliance is an umbrella organization of all compost organizations in the world, so from Canada, United States, New Zealand, and Australia. And today we have set up a series of different webinars. So you have the possibility to join the next webinar, which is organized by the International Solid Waste Association. And I'm sure Jane Gilbert, she will present the new report, the contamination report, preventing and managing contaminants in organic recycling. So join this at 1 p.m. And you have the chance as well to go at 4 o'clock to follow the 6 C's approach of the Compost Council of Canada. And if you are still chilled, then you can go in the evening to the Compost Research and Education Foundation to deep dive into the role of compost stormwater media. I would like to thank all participants to be on board today on our webinar, and I wish you a nice World Soil Day. Thank you very much and goodbye.